Hello, my name is Ashley, this is Faithful Rattos, and welcome back to my channel. So we're going to do another one of those slightly complicated genetics videos today. I want to preface this by saying if I do make a mistake with the genetic lingo here, please do correct me in the comments, that's totally cool. This one will get a little bit more complicated than the previous one. This one is talking about codominant things-ish and a little bit more complex ideas. So if you haven't watched the videos before, do go and check out my video that I made on the agouti gene. That should clear the basis of genetics up for you. So today we're going to be talking about a specific loci, and a loci is just like a place where you find the genes, where you find the alleles, and we're going to be talking about the C loci, and the C loci is associated with albino rats and shaded rats. So we're going to talk about a couple of the different alleles that can be present on this loci, what they do and how they work with each other and things like that. So we have the dominant, which in this case is a capital C, as in the C loci, and this is the switched off. This is the off version, and the on version has a lot of different variants. So there is the lowercase c, and this is the albino. This causes the black and the yellow pigment in a rat's fur to go down to white, so then you'll get a white pink-eyed rat, an albino white rat. Then we also have the CH gene, which is like a lowercase c with a little h above it, and that is the Himalayan gene. We also have two genes that I'm not going to talk about very much today. We have the C little m, the little c tiny little m, and that is the Martin gene. Now the Martin gene is very interesting. I'm not very clued up on Martin. It's quite a relatively new variety, so I'm not super clued up, so I'm not going to be talking about Martin. Himalayan gene, the little lowercase c and the little h, you have one copy of that and one copy of the albino gene, which is just the lowercase c by itself. Together these will produce a Himalayan rat. If you have two copies of the c lowercase c little h, lowercase c little h, that will produce a Siamese rat. And then from there, the shaded pointed varieties get ever more complicated with ever more dilutes and extra recessive genes that you can add on top of that. So the lowercase c little h gene does something interesting, which is called acromelanism. Now albinism, I think most people are aware, is where there is no pigment at all. But acromelanism causes something very interesting to happen. So this gene causes albinism, but at a certain temperature is switched off. So the extremities, the colder parts of the body, do not have this albinistic characteristic which creates the points. So the coldest parts of the bodies, the nose, the ears, the tail and the rump leading to the back of the body, will have more pigment than the rest of the body, and this leads to the shadedness about the varieties that we're going to talk about. So they have the nose point or the face point, they have the point on the ears, on the feet, on the hands and on their tail. This type of gene is present in a lot of animals. It's present in guinea pigs. You have the California and Himalayan guinea pigs. It's present in rabbits. It's present in cats. It happens in a lot of species. It's one of those genes, as I mentioned with the agouti. It is present in a lot of different species and it does some really cool things. We've talked about Himalayan and we've talked about Siamese. I think a lot of people will be wondering, what about Burmese? Now there's a lot about the Burmese genetics that is as of yet unproven. However, it's basically a modifier on top of this base we already have, which is lowercase c h, lowercase c h on the C locus, and they call it the Burmese gene. With one copy you get a Burmese, and with two copies you get the sable Burmese. Now the Burmese is a nice chocolatey brown and the sable is darker again, and you get this sort of otter, I think they call it, shade of brown with the points similarly in the same tone as the base colour of the rat. Here's where we can talk about different flavours, as I mentioned with the different agouti based rats. A lot of these different modifiers and dilutes will impact how a rat looks. So if we add agouti into that, the agouti Siamese will have a much less pronounced point. The standardized variety of Siamese, at least here in the UK, is based on an lowercase a, or better known as a black base, but you can have agouti point Siameses. They're just a little bit paler and they don't have as much shading. Then if you throw the agouti gene into a Burmese, you get something called a Wheaton, which is an absolutely beautiful variety. One of my favourite things. Eventually, when I'm able to start breeding, I think Wheaton is one of the, I just love them so much, is one of the ones that I'd consider depending on the available rats at the time and things like that. Wheaton is absolutely gorgeous. They get the heathering that agoutis get. It's slightly lighter than the Burmese, but it's so pretty and I absolutely love it. Then we can have also any of the other modifiers on top of that. So a very common modifier to find these shaded varieties in is the Russian blue gene. Now the Russian blue gene will create a blue point Siamese. So instead of this like rich, like sepia toned point, the Siamese will have sort of like a bluey grey point. Really pretty variety. For the Burmese, this creates the Russian Burmese. I have a Russian Burmese. This isn't quite standardized here in the UK. It's in guide standard, but my Bentham is a Russian Burmese and I think they're super, super pretty. Then we also have something that is even less standardized than the Russian Burmese. You have a Russian Wheaton. This is my absolute all-time favorite variety. The most special rat to me, who you guys probably know, Plum. He was a Russian Wheaton. It's not standardized in the UK, and a lot of the reason why a lot of varieties aren't standardized here in the UK is keeping on top of those and keeping judges well informed about all these different varieties is very difficult. So when there's varieties that look very similar and it's very hard to properly distinguish them, this can hinder them getting a standard, and that's the case with Russian Wheaton. As on top of that, not many people breed it. I don't think anyone breeds for it. There's a couple of breeders who will have it out, but nobody actually breeds for it, so it's not, you know, common, and so it doesn't have a standard. But I think Russian Wheaton is so pretty. And also onto that, we can talk about the golden variety, so golden Siamese, golden Wheaton. I don't think anyone's played around with a golden Burmese. They might have done, they might have had one out. I don't know how that would affect it, but this creates a really sort of rich, warm-toned version of these colours. And then there's one thing that is kind of, I personally would lump into this shaded pointed like category, although I'm not sure if everybody would, and that is the Tonkinese. Now Tonkinese is much more improving again than Burmese. From the conversations I've had with breeders who are working with Tonkinese at the moment, it works on a basis pretty similar to Burmese, where you have the Burmese gene with one copy creates an effect, and then two copies, or homozygous. Homozygous just means two copies of the same allele, will create like an even darkened version. So in my eyes, it works a bit similarly to the Burmese gene, creates a really interesting look of rats, and people are playing around with like Russian and agouti versions of this gene. Very cool, very interesting, but all under this little shaded pointed like category, and it's all because of that little lowercase c little h gene. And it's so interesting. I love shaded varieties. As you can tell, they are my absolute favorite. I'm a bit biased when it comes to shaded varieties. I absolutely love them, specifically Russian Burmese and Russian Wheaton, but Siamese has a really special place in my heart as well, and I do love a good Burm. But yeah, so that is all about the sea locus, the pointed shaded varieties. As I mentioned, I don't think I have enough knowledge when it comes to the other shaded sea locust varieties, the Martin, but that's not to say in the future I won't, and that I could make an addendum to this video. So yeah, that is the whole video. Again, another little shorter video talking about genetics. I know this isn't everybody's thing, but as you can tell, I get really excited when it comes to rat genetics, and I find it very fascinating. And as somebody who is looking to become a breeder at some point in the future, when I feel a little bit more, I think, financially ready to take on that many rats, that you need to have a proper breeding situation. My 10 boys are about much as I can afford, so having 20 or more rats for a breeding program just isn't feasible for our situation right now, but at some point in the future it will be, and I will be so excited. But I do think getting clued up on rat genetics is really helpful for that day when I do want to become a breeder, because then I have all the knowledge already. Also, it's fascinating and I absolutely love it, and anyone who will spend at least 10 seconds with me will know this. I'm gonna shut up because this is quite awkward. Why do I say things like this? That's all from me and my blubber mouth who won't stop talking about rat genetics, because why do I find it so interesting? Who knows? Anyway, I'm gonna take my awkward self off this video because nobody needs to see how much I've had to cut out of this video. But yeah, thank you very much for watching. I really hope you enjoyed this video. Maybe it was a little bit informative. If there's a specific gene or variety or something you think that you would want to see a video on, do leave that down in the comments. I'll try my best to do a little bit of research and get back to you on that. Thanks very much for watching, and I'll see you in the next one. Bye!